The Historical Component of Recombinant DNA Technology by Amber Myers. In this video, I'll be providing a comprehensive analysis of the CRISPR-Cas9 recombinant DNA technology and placing an emphasis on the historical perspective to underline the aspect that this was an ensemble endeavor. It was Carl Linnaeus who said it best with, "The first step in wisdom is to know the things themselves." So what do we know about CRISPR-Cas9? Well, it stands for Clustered Regulatory Interspace Palindromic Repeats, and it's a system that was discovered in ancient Archaean bacteria. It's a built-in immune system that is flanked on either side by catalytic enzymes, which are able to cleave any bacteriophages that invade. This technology was able to be harvested and used in the COVID-19 vaccines. We'll talk more about that in the next slides. So why is the historical perspective important in understanding recombinant DNA technology and modern vaccines? Well, if you think of it as a series of stairs, you can't get to the step ahead without first passing over the step behind. And sometimes these steps need tweaking, but sometimes there's definitely experimental evidence under the foundation of those that helps build upon the next idea. So it's important to understand that this technology was an ensemble endeavor and that there are scientific minds that helped to produce it before those that were noted in 2020. So we're going to talk about those in the following slides. So let's talk a little bit about the development of cell theory. Before the cell theory, cells were not even really considered individual living units, but were just thought of as a network of channels. The first two cell tenets were developed by Theodor Schwann and Matthias Schleiden. Schleiden was the one who worked on plants, and Schwann was able to apply his applications to animals. Cells were not even considered living units, and it was Robert Hooke, the first individual who discovered cells, who actually thought of cells as a network of channels. Let's take a look at the history of cell theory and some of its important researchers, because without them, these important modern breakthroughs in recombinant DNA technology could not have occurred. Each cell leads a double life, an independent one, pertaining to its own development alone, and another incidental, insofar as it has become an integral part. So both of these men wanted to understand the cell more. Matthias Schleiden worked on plants, and while he was able to understand a lot of the individual components of plants, Theodor Schwann was able to take these applications and use them with his research in animals. He used a lot of animal tissues and fluids and was able to study those. Because both of these men believed that cells were to be thought of as not only a collection of of cells, but also individual units. That is the most important thing to remember here is individual units. They're composed of, one or more cells. All organisms are composed of one or more cells, and that the cell is the basic unit. So in all life, there are cells. That's basically the first two tenets, and these guys are responsible for that. So that is important in where we are today, and we're going to move on to the third tenet. Next, I want to talk about an important discovery in 1855, that the third tenet of cell theory expressed that cells have to arise from cells which already exist. This weak to the process of nuclear division was something which was unforeseen before their time. This important element leads to the understanding that mitosis is the process of cell division. Robert Remak and Rudolf Virchow were extremely interested in biology. They were interested in diseases, cytology, and embryology. It was their important discovery and observation based on the nuclear division and the cleavage of the zygote in germline cells that helped provide the foundation for scientists such as Jennifer Doudna to be able to understand how DNA divides. Her work with mRNA was paramount in the research that contributed to vaccine development for the COVID-19 vaccine, which we'll discuss in further slides. Now landing on Mendelian genetics. The father of modern genetics, Gregor Mendel, was born to a poor family of farmers. His lack of influence and money really prevented his ability to be able to establish support for his ideas. He even had to drop out of college and go to the St. Augustine Thomas's Abbey in Brno. His ideas about particulate inheritance, as well as his laws of segregation and independent assortment, were very important aspects in Mendelian genetics, and we still use them today. I wonder what would have come of his ideas if he had never sent that paper to Carl Nageli. All right, I want to talk now about the bacterial transformation studies, and why they are historically important is because the understanding of the gene regulation and negative regulatory mechanisms that are found in archaea and bacteria really helped to catalyze the knowledge base that was, the framework that was put in place for future scientists to look at bacteria and understand how the immune processes inherent within single-celled organisms can actually be harvested and used for the benefit of eukaryotic organisms. So first we're going to talk about the pajama experiments, which involved Arthur Pardee, Francois Jacob, and Jacques Monod. These experiments were important, and they did involve mutations in the E. coli. And that led to the discovery of the lac operon, which involved beta-galactosidase and lactose, and the negative regulation mechanisms, the gene regulatory mechanisms that were discovered because of that. These ideas were able to be applied to future studies, the Britton-Davidson model, which was the first eukaryotic gene regulation study performed by Roy Britton and Eric Davidson. In 2002, Eric Davidson was able to publish a diagram that actually showed interactions in a network of over 40 genes in sea urchins. So that was a really important discovery. So it all builds up to genome editing, and why is genome editing important within historical applications? Well, starting in the 1970s with Paul Burt's recombinant DNA and Asilomar 1 and 2, we were able to see how lack of transparency within the scientific community and public sentiment can lead to problems. The SV40 phages and their association with tumor-causing properties is definitely an issue that everyone should know about. In '86, Yoshima Ishizunu, doing his PhD research in Japan, was able to first identify CRISPR, but he didn't know what it was, and he didn't give it its name. He just basically called it out as a series of repeating sequences, and that was that. Between '92 and 2004, Francis Mojica, the Spanish scientist, was able to discover CRISPR once again, and he gave it its name. He knew it had to be important because DNA on bacteria is so highly conserved and already in such small numbers compared with the base pairs in eukaryotes. So if something is repeating that much and that often, he knew it had to be important. He tried and tried again and again to have his paper published. I know several of the journals turned him down because of his lack of influence. One of them was Nature, but the Journal of Molecular Evolution finally published it in 2004, and this was how other scientists were able to really start understanding more about CRISPR. Jennifer Doudna in 2005, along with Gillian Banfield. In China in 2018, unfortunately, Dr. He Jiankhu was able to use genome editing tools on the germline cells, which caused a lot of issues, and he is in prison for that for 3 years. He might be getting out this year. In 2020, Jennifer Doudna, along with Emmanuel Charpentier, were awarded the Nobel Prize for their work with CRISPR-Cas9. And just recently in 2020, Jennifer Doudna and her team of colleagues at UC Berkeley worked tirelessly to push this technology into the hands of other pharmaceutical companies. The COVID-19 vaccine and mRNA technology was developed with Moderna and Pfizer-BioNTech under the Emergency Authorization Use Act. And the biggest hurdle that we still face today is public sentiment and public confidence. Thank you for listening, and I'll leave you with an image of an empty workbench. You know who it is.